So first of all, I just want to thank the organisers and the hosts, Zespri International and the Riddet Institute for giving me the opportunity to be here today and talk to you about the evidence and the research that has been done in vitro and in vivo to support a role for kiwifruit in digestive comfort. Secondly, this work couldn't be done by itself, and so with this slide I just want to acknowledge the key people that were involved in most of the research that I'll talk to you about today, mainly based at the Riddet Institute at Massey University in Palmerston North. It's important to note that a number of other people were involved both around New Zealand and internationally, and so they will be referenced where possible. Okay, so digestive comfort. What does it mean? Why are we talking about it? Why is it important? Well, hopefully you all feel nice and comfortable in your digestive system after eating lunch, and it's definitely the preferable feeling to have after we eat. But one of the important things about it is that it gives us an indication as to the health of our digestive system, which is important to be able to gain all of those nutrients and energy out of our food. And so digestive discomfort can indicate anything from a mild disturbance right through to the diseases that we've just heard so much about. Now, there's a number of factors that are associated with digestive health, but I'm going to be talking to you about two of those in most detail today. So we have digestion, and then we have laxation, or our bowel movements. Digestion involves the physiology of the gut, so the structures that make up the gut and how they work together. The gut microbiota, so the bacteria that live in our gut and play important roles in digestion and in producing certain metabolites, as well as enzyme function. So these are the enzymes which are responsible for digesting our food, such as proteases that digest protein. Now, digestion is important for laxation. Laxation involves the characteristics of the stool, such as how soft it is, how much there actually is, as well, the stool bulk, and also the gut motor, or those muscular contractions that occur to push food through the digestive system. Now, in terms of the latter, the gut transit time, that's the amount of time it takes for food to actually move through. Perhaps the most important region of the digestive system is the large intestine because, as you can see, food spends most of its amount of time here. However, the stomach and the small intestine, the upper tract, in here we have a lot of the dynamic processes of digestion that occur, and so this is also very important. And so I've showed you the key factors involved in digestive health, but what does digestive health actually look like? How do we know that we have both efficient digestion and optimal laxation? We want to have a soft, bulky stool and frequent bowel movements, and an optimal transit time, both the absence of constipation and diarrhea, and you've heard a lot about that already. We need to have a healthy composition of gut bacteria due to their important roles and the metabolites they produce, such as short-chain fatty acids that I'll talk a little bit more about. We need a healthy mucus layer. This is the lining of the gut, and it plays an important protective function, so it needs to be intact. And then, of course, we really want that digestive comfort, okay? The absence of bloating, pain, and excessive flatulence. And so what we're really interested in, and what I'll be talking to you about, is how does kiwifruit promote all of these outcomes? Now, kiwifruit are well known to promote laxation, and we just heard about clinical trials that were done and some promising results from an ongoing trial that's underway. And laxation, and there's the bowel movements and those kind of things, they're important endpoints to measure because they tell us a lot. But what we really want to know as well is what are the mechanisms that are involved in this endpoint? How do we get through the digestive system? How does kiwifruit do what it does? And we also want to know about digestion. How does kiwifruit impact that? Now, in order to understand these mechanisms, we turn back to model systems. A number of model systems are used to investigate those physiological processes in more detail, because there's a lot of things we can't actually do with humans. And so I'm just going to tell you about the in vitro and in vivo models that we're going to talk about mostly today to study digestion. So in vitro, we use a jacketed reactor system. This mimics the chemistry of human digestion, and it carries it out at 37 degrees Celsius. It's got two phases. The first phase is the gastric or the stomach phase. There's a low pH, pH 2, simulated juice containing pepsin, the stomach enzyme is used, and that's carried out for 30 minutes. The pH is then bumped up to pH 8, so that's going to simulate the small intestine. The simulated juice now contains pancreatin, an intestinal enzyme, and that's carried out for another 120 minutes. So that's the in vitro system. It gives us an indication of what's going on. But then we want to turn to some in vivo systems. So that's going to allow us to study things at the whole organism level. The first in vivo system usually used is the rat. This is because it's easy, it's inexpensive to use, but it does have a lot of digestive differences to the human. And so if we want to use the best possible model of human upper tract digestion, we use the growing pig. And this is a pig around 25 to 60 kilograms. Like humans, the pig is a meal-eating omnivore, and so that's one of the reasons why it's such a good model. Now, what does the research say? So, laxation effects have generally been attributed to fibre. We know that kiwifruit is a good source of fibre. About 2 to 3% of the fresh weight of kiwifruit is fibre, and green has more fibre in it than the gold. This fibre is both insoluble and soluble at a ratio of around 3 to 1. And it's got a number of important properties. Okay, so hydration properties, we have swelling, a high water holding capacity. Undigested fibre will contribute to the bulk of the stool, so there's more there to actually pass. And fibre is also fermented by our gut bacteria to produce short-chain fatty acids, as well as some other metabolites. Fibre also has roles in diffusion and mixing, but this is more relevant to metabolic health, and that's going to be covered tomorrow, so I won't talk any more about that. What I'm going to talk to you about today are hydration and bacterial fermentation mainly. So swelling, this is the volume that is actually occupied by the fibre, and water holding capacity is the amount of water that's tightly bound to the fibre as it passes through. And what you can see is underlined for both green kiwifruit and gold kiwifruit that this kiwifruit has high levels of both swelling and water holding capacity compared to other sources of dietary fibre. As I said, undigested fibre will increase the bulk of the stool, and so all of these things are mechanisms that improve laxation. Now, kiwifruit fibre also acts as a prebiotic. So prebiotics are things that selectively promote the growth of good bacteria in the gut. So this is such as the lactobacilli and the bifidobacteria species. This is important because the gut microflora have a key role in digestive health, and one of the ways they do this is through the development and promotion of gastrointestinal motor function. So those are those muscular contractions that occur. The microflora also ferment the dietary fibre to produce a number of important compounds. One of these groups of compounds are short-chain fatty acids, and these have been proposed to stimulate the gut motor. And those short-chain fatty acids are also an important source of fuel for the colon cells and also other good bacteria in the gut. But also produced by the gut bacteria are polyphenols, which may inhibit the growth of unwanted bacteria. And so this is great because you have both the promotion of the growth of good bacteria and the inhibition of the growth of unwanted bacteria. A number of studies have also demonstrated an association between gastrointestinal disorders such as IBS and an imbalance of those microbes. So in this in vitro study, what was done was gold kiwifruit and green kiwifruit were compared to inulin, which is a known prebiotic, so it's known to modulate the gut bacteria, and this was compared back to a no treatment control. And so what you can see is that both gold and green kiwifruit are able to modulate the gut bacteria at a comparable level to inulin, the known prebiotic. But in the same study, the production of short-chain fatty acids, butyrate, acetate and propionate was also measured. And butyrate particularly is interesting because it's been suggested to promote that motor function via stimulation of the gut's nervous system. Again, what you can see in the graph on the right is that compared to that known prebiotic inulin, both gold and green fibre were able to stimulate the production of good levels of butyrate. Now, in addition to the polyphenols that are produced by the gut bacteria, kiwifruit itself may also contain polyphenols. So what you're looking at here is the ability of a range of different polyphenols to inhibit the growth of salmonella, so that's an unwanted bacteria in the gut. And those polyphenols that are contained within kiwifruit have been highlighted in green. Another thing about kiwifruit fibre is that it may stimulate mucin production in the stomach and the duodenum. So mucin production tells us about that mucus layer that I said was so important for digestive health. It's important because it sits on the gut wall in between the gut internals and the host, and so it's pretty much the first line of defence from environmental pathogens. Its function depends on the thickness and the rate that it can be replenished. And again, a damaged mucus layer is associated with colonic diseases. So this study was done in growing pigs, and they were fed increasing amounts of dietary fibre. And after feeding, mucin production and also the expression of genes associated with mucin production was measured in different regions of the digestive tract. And what you can see is that as the amount of fibre in the diet increased, the production of mucin also increased. And this was found in both the stomach and the duodenum. In addition, the expression of genes associated with mucin production in those locations also increased significantly for the duodenum. Now, what are the mechanisms that are associated with this? It could be direct, so fibre may have an abrasive effect, breaking down the mucus layer, and in breaking down the mucus layer, it stimulates the replenishment and so the production of more mucins. But also indirectly, because fibre has that role in modulating the gut bacteria, and the gut bacteria also have a role in maintaining the mucus layer. Further research to understand this in more detail is underway. Now, I've showed you about fibre and its impact on laxation, but fibre can't take all the credit for laxation, and also fibre can't take all the credit for all of the digestive health benefits of kiwifruit. So, for example, other components of kiwifruit have also been suggested to affect mucin production: actinidin, polyphenols, and raphides. Actinidin, you've heard it mentioned once today already, I think. It's a protease, that's an enzyme that digests protein, and it's been suggested that this may just help to promote the natural degradation of the mucus layer and therefore stimulate its replenishment. Like fibre, the polyphenols that I've told you about also modulate the gut bacteria. And then raphides are small, insoluble, needle-shaped crystals that are contained in kiwifruit, and these have been suggested to have a stimulatory effect on mucin production. But what I'm going to focus on now for the rest of my talk is actinidin. Actinidin is predominantly in green kiwifruit, but SunGold kiwifruit contains about a quarter of the level that's in green. And the original gold kiwifruit, which is Hort 16A, had no actinidin at all. And so for this reason, often the original gold is used as a negative control in our experiments. So a little bit of history about actinidin. The presence of some kind of proteolytic enzyme in kiwifruit had been known about for many years. For example, jelly won't set if kiwifruit's added, and kiwifruit, when placed on meat, has a tenderising effect. And then in 1959, actinidin was actually named actinidin, and in the 1970s, the enzyme was purified, and details about its mechanism and structure were elucidated. Now, as I said, actinidin is a protease that digests protein, and efficient protein digestion is an important part of digestive health and digestive comfort. So meals high in protein, though, can contribute to gastrointestinal discomfort. So you'll know that sometimes if you eat a meal with a lot of meat in it, it can sit in your stomach and it can feel very heavy. And so the addition of proteases in the diet may help with this to assist efficient protein digestion, and this may contribute to gastrointestinal comfort. So we wanted to ask, does kiwifruit have a role in protein digestion in the stomach and small intestine? And so in order to answer this question, a comprehensive series of in vitro and in vivo experiments were designed and carried out. And that's what I'm going to talk to you about now. So first of all, in vitro. In vitro, what we did was measure the digestion of a range of different food proteins, both with and without a green kiwifruit extract. And what you can see is the two phases there in vitro, the gastric phase and the gastric plus the small intestinal phase. For the gastric phase, casein, meat protein and soy protein all had the digestion significantly enhanced when actinidin was added to the system. For whey, zein, gelatin and the wheat proteins, there was no enhancement. But then when the small intestine phase was added, all of those proteins were significantly enhanced. The digestion was significantly enhanced when kiwifruit was added. Now, in order to gain an indication about what's happening with digestion visually, we use a technique called SDS page. Every treatment occupies a different lane of the SDS page gel, a different vertical lane. Every one of those kind of purple bands on that gel represents a different protein. And the density of that band, or how dark it is, tells us how much protein is actually there. And the vertical position that it is on in its lane tells us how big it is. So if we look at the main isoforms of casein, we can see that they're at the top, the top of the position in the lane. That tells us that they're big, they're undigested. And there's also a lot there, the very dark bands. If we then go to the next lane, this is casein plus pepsin, so it's the simulated juice but without the actinidin, the green kiwifruit extract. You can see that we still have bands at the top, so some has been digested, some undigested, and you've also got some bands appearing down the bottom, and that showed us that indeed some digestion has occurred. However, if we now take the casein and we add actinidin to the mix, we can see that those bands that were at the top of the gel have virtually disappeared. Okay, so that's telling us that actinidin has significantly enhanced digestion of the casein protein. So now we move into our in vivo model system. So first of all, the rat. So rats were fed diets containing specific proteins along with either the green kiwifruit containing actinidin or the gold Hort 16A kiwifruit without actinidin. Once again, we have an SDS page gel. So along the top, you can see the different proteins, and then for each protein, there are 3 lanes. The first is the diet only, so that's no digestion. The second is minus actinidin, the gold without any actinidin. And then the third is plus actinidin, the green kiwifruit. Very simply, what you can see is if you compare the minus to the plus, you can see that for beef muscle, gelatin, gluten, and SPI, which is the soy protein, there is an enhancement of digestion when actinidin is added. And those numbers relating to that are shown down the bottom. However, for whey protein and zein, there's no effect. Those 3 lanes look very similar to each other. Right, so the other thing that we can measure in rats is something called stomach emptying, or the stomach emptying rate, and this is the rate at which food will go from the stomach through to the small intestine. And we do this using a very elegant technique called wide-bore nuclear magnetic resonance, or NMR, and this allows us to measure stomach emptying in real time. We do this by adding a naturally occurring aluminium isotope to the diet. This is 27 aluminium. And we can measure this because this isotope of aluminium is only detected at a low pH. So when it's in the stomach and the pH is low, we can detect it, but as it moves out of the stomach to a higher pH, it's no longer detected. And in the chart on the right, what you can see is that peak of 27 aluminium decreasing over time. Now, what we're actually measuring is something called T-half. This is the amount of time required for half the stomach contents to be emptied, and what it's telling us is that a smaller T-half reflects a faster stomach emptying rate. So what you can see for your dietary proteins is that for beef muscle, that the T-half was significantly decreased when actinidin was added, but although there was some change for gluten, soy and whey proteins, that wasn't a significant change. And if we look at the kinetics of that on a chart on the right, that is consistent with those numbers. Now, this increase in stomach emptying rate by actinidin may be related to the effects of kiwifruit on gut transit time. So we turn now from the rats into our growing pig model. You remember that's the best model that we have for human upper tract digestion. And because we saw such promising results with the beef muscle protein in the rats, we use that in the pigs. So pigs were fed diets containing the beef muscle protein together with, once again, either the green or the gold kiwifruit, the gold Hort 16A. If you look at the SDS page on the right, and we focus on our Hort 16A, what you're looking at is the time of digestion, and you can see that at 0.5 and 1 hour, it looks very similar to the lane that says diet. Okay, so not much digestion has occurred. It's not until about 7 hours that digestion is complete. However, if we look at the green kiwifruit, what you can see is that if you compare the 3 hour for green to the 7 hour for gold, they look very much the same, showing that green kiwifruit containing the actinidin has enhanced the digestion of the beef muscle protein in the pigs. In terms of numbers, this means that gastric digestion was increased about 50% with the actinidin containing kiwifruit. We also measured stomach emptying rate, and this was there was about a 20% decrease in T1/2, showing a faster stomach emptying rate with the green. It's also important to note that, like human digestive enzymes, actinidin is active at that low pH over the time course of normal digestion. Now, to follow these up a little in humans, a clinical trial was conducted to look at how actinidin might contribute to gastric comfort after a high-protein meal. So 10 healthy adult males were fed meals with 400 grams of lean steak, either with the green kiwifruit containing actinidin or the Hort 16A gold kiwifruit without actinidin. And in order to monitor this, questionnaires were filled out, so they reported a number of different measures of gastric comfort. And for every single measure, the direction of the results was the same. When green kiwifruit was consumed with the high-protein meal, there were less symptoms reported. And for reported bloating, this was significantly less when actinidin was consumed. Okay, so I've showed you a lot of different research around the possible mechanisms for kiwifruit's effects on digestion. What does this mean in terms of our model? Kiwifruit has a good amount of fibre in it. Fibre has properties of a high water holding capacity and swelling, as well as providing bulk that promote laxation. Fibre also modulates our gut microbiota, which produces our short-chain fatty acids that also have a role to play. Kiwifruit also contains polyphenols, and these also modulate the gut microbiota. Fibre also positively impacts the mucus layer, and the microbiota may also have a role in this. I also told you about actinidin. It increases protein digestion and the stomach emptying rate, therefore enhancing the digestion of proteins. Actinidin and raw feeds may also have a benefit to the health of the mucus layer. So your take-home message from all of this in vivo and in vitro evidence is that cutting-edge evidence shows that kiwifruit enhances digestive health and comfort. Kiwifruit is well known to promote laxation, but fibre is only one part of that laxation story. And then beyond laxation, there are many other digestive health benefits, including protein digestion, prebiotic activity, and mucin production. Lastly, I just need to thank Lindley Drummond and Juliet Ansell for further research support. Thank you very much.